letters. So dot plots are plots that we make and we put the dots wherever there is something that coincides, and you'll see what that means. But as usual, here I'll start with what do I want you to learn from this? Okay, what are dot plots and what do we use them for? And so they are graphical representations of an alignment, and we use them to see alignments more easily. You'll see what I mean by that. How does the computer generate those? You're going to do this by hand. That's why I told you print this file. Then you're going to learn what the patterns mean, which is a question that I normally ask in homework and in exam. I give you the result of a dot plot, and I ask you, what does it mean in terms of DNA sequence? Because the different patterns that they make, they carry meaning. And it's kind of fun to be able to look at the pattern and know what it means in terms of sequence. Okay, then increasing and decreasing window size. So you're going to learn, you can either compare one-to-one or you can compare pairs of things. So we're going to do a little bit of this today and a little bit of it Monday, which then you also bring me questions about for review. But we have one more class, that's the running Dotplots, the bioinformatics tool. And some of these I already alluded to. So the gaps and the indels, they actually are the same thing. Indel is short for insertion, deletion. Insertion, like, okay, there wasn't this letter here, this nucleotide, but now there is. And deletion is, there used to be a nucleotide or a chunk of nucleotides here, but now there isn't. Since we don't know which one came first, just call it indels. Gap is the other terminology for indels. And then inversion is a break on the DNA sequence, and then it flips on itself and then rejoins. You will see what that looks like in terms of sequence. The duplication is, there is this DNA sequence that will appear somewhere else on the genome, and maybe it's intended, like one and then repeat itself right after it, or it could be somewhere else on the genome. And the window size is you're going to see what happens when you increase or decrease it. So this is what a dot plot looks like. And what you do is you write the two sequences that are aligning. So remember, an alignment is when you line two things up and you're comparing them and you're seeing what they have in common. So you're comparing this sequence with this is a sequence. And so what you do is you go down this line, T matches T, oh, dot. T doesn't match H, no dot. T and I, no dot. So you keep going like, and then like, okay, only this one matched. Move on to the next one. So this is showing you a window size of 1 because it's just comparing 1 to 1. Sometimes you want to do like, how about, does TH resemble TH? And then you just put one dot. And there are reasons why you'd want to do that. But at this point, let's just go with the window size of 1. That's the simplest scenario. I don't know if you can already see some pattern in here. Like put on the chat, when you look at the red dots versus the pink dots, what do no, one of them is special. Like which one is the special and why is it special? So as a hint, you're looking for patterns. Right, the red makes a pattern. Good job, guys. So, right, so what you can see, perfect, exactly, Leandro, that the red, they are lined up. See, they make this diagonal. So why did the computer make this in red and this one is in pink? Because they want to show this is a more meaningful match than this. Okay, so you're like, okay, S and S match. S and S, they also match. But this S here doesn't actually line up, like doesn't make a diagonal with that. So this could be what's called a spurious match. Like it's a match that is not as relevant. It's like, I'm looking for my soulmate. Okay. I don't know. There are lots of characteristics that are important. And then here, I like water. He likes water. Okay, whatever. So it's a match just because S is a very common letter. So that S is a better match with this S than with this other one because it makes a diagonal. So what we're looking for is we want to see lines, contiguous lines. They are going to be either continuous lines, and you will see when are they continuous lines, or they're going to be continuous and then flipped, or they're going to have this breaking here. They each have a meaning. So here I just one day had a lot of time in my hands, and then I thought, let me show them what happens when you're comparing DNA. So I put that sequence there. So this I am aligning them. I'm aligning this one and this one. And hold on, quit this thing. Okay, so I'm aligning both of these. And if you're going to not make a dot plot, you're just going to go like compare each two. But the dot plot helps you develop that pattern. And here I am cheating a little bit, you will see. So I'm only comparing that A with the A. And when I said I'm cheating, I'm already telling you that I just said, dot there. And then I went, and I didn't put a dot there. At one point, I'm not going to cheat, and you'll see what happens if I don't cheat. You might already be predicting, but you will see. And I'm like, okay, next letter, next letter, next letter. And I'm like, oh, okay, this here. So the reason I made this in a different color is to show you that it's not forming that line. But it's going to form some interesting line. Like, what's happening? And then back to blue. And so when you look at that, the reason why I made this dot plot is to show you there's a contiguous line, another. So the diagonals, they're showing to you that these parts are the same, they're similar, right? So AGC and AGC. And this other blue here and on the bottom is this and this. Like, oh, same sequence. They are forming that diagonal. And then this one is diagonal the other way. And now if you compare these two sequences, what do you think that made this diagonal not to be straight, but like flip like this? What compare this to that? Yeah, so then you said it's an inversion. Yeah, so how did he see that? Okay, GGATT, and it's an inversion because if I read from right to left, is an inversion in here. So remember, we always read 5' to 3', but it doesn't mean that the chromosome doesn't break and does like a headstand or whatever. So this breaking that line and with an interrupting line shows you an inversion. So that's so far you've learned about two patterns. The one that is just a diagonal, which shows you similarity. The diagonal that is interrupted by a line that is orthogonal to your main line, that's an inversion. And then there was going to be another pattern that you will see. But I think that's when I'll do the exercise. No, here's when I'm going to show you when I don't cheat, then what happens. And when I say when I don't cheat, initially I was comparing 1 to 1, but not actually putting every dot there. Now it's when I'm being more honest and putting a dot where every time one letter matches, I put a dot there. Then it's like, oh, that T matches a bunch. It's not just that one. And then I did it for the next one, and I kept going and taking a bunch of screenshots. I'm like, oh. So I get this. And then I'm like, okay, do I see any diagonals here? And if you look at the dot plot there, it's just kind of like, well, maybe a pattern. Maybe I see a main diagonal, but these two sequences that I'm comparing here are the same two sequences that I compared over here that had that inversion. Oh, no, wait, I think it changed. Yeah, okay. I should have done the same sequence, but all right, so here is a different sequence. So I actually cannot, I don't really know what pattern is hiding in here. And the reason why they matched so often is because there are only 4 things they can be, right? In each position, it's either A, T, C, or a G. So there will be lots of these spurious matches, or we could say that there's a noise. So the noise is the signal that comes on top of the signal that matters, right? We want to, the signal that matters is whatever is going to make a pattern. And there are so many matches that it's hard for me to see what the pattern is. So I don't know how similar they are. All right, so the reason for that is because 1 in 4, very frequently you could have that match. So any ideas how we can decrease the noise? So you could say, well, let's compare protein instead, because protein has 20 different possibilities instead of 4. So there are 20 different amino acids. For DNA, there are only 4 possible nucleotides. And for protein, there are 20. And I'd say, okay, but what if I still want to compare DNA? We can't increase the number of nucleotides that exist, but what we can do is increase the window size that I'm comparing. And when I mean window size, and I don't, you guys see my mouse? And then wait. Ah, yes, now you do. When I say window size, instead of comparing only T to T, let's say I make a window size of 2. So I'm going to do like, I'm going to compare TG. Let's see, how, which block out. So we're comparing this strand to this strand. And this dot plot here looks really crazy because there are lots of matches. And now I want to know, are there diagonal lines here that are hiding in this mess? And then what I'm going to do is increase the window size. But I think Caitlin has a question about the previous plot, so let's go back. Let's see, the inversion is GA, maybe, let's see, maybe that escaped me. C, no, the inversion is GGATT, GGATT. So that C stayed there, then, okay, all right. So here on this one, that looks really messy. And I was telling you that there is a lot of noise because the window size was small, and the chance of matching 1 in 4 is a high chance because there's not a lot of variation. So now we are going to say, let's compare 2 of them, and we only put a dot whenever 2 of them match. So we are increasing the window size. So, okay, this is some math because I might like math. If you're comparing 1 to 1, your chance of matching is 1/4. If you're comparing 2 nucleotides, then you have to match the first one and the second one. And when there's an and, you multiply that, so your chance of a match is 1 in 16th. Let's see, on the 4 or 5, let's see. 1, 2, 3, 4. 1, 2, 3, 4, 5. Yeah, where did I miss? I think I missed a red dot somewhere. So, oh yeah, okay, here. I guess I skipped that one. Wow, you were meticulous, Caitlin. You should go into bioinformatics, because when you're programming, if you make this kind of mistake, your thing doesn't compile, it doesn't run. Okay, so yeah, I'm missing like 1 red dot here. Good catch. I came up with this exercise like 8 years ago, and I never noticed. Nobody ever noticed. Good for you. I did it on purpose to see if you're paying attention. Okay, so back to the window size, right? We want to decrease how often two things match just by chance. So we're going to compare two nucleotides instead of just one. So for us to put a dot, there's a 1/4 chance, and so for the first nucleotide, and a 1/4 chance of the second nucleotide. So now my chance of match is 1 in 16 rather than 1 in 4. So here's the window size of 1. That's the slide I had shown earlier. Here's the word size. So some bioinformatics tools call it word size, and it's called a sliding window. So what I did here, I could have skipped this one, but let's say I look at a TG and TG, and if they match, I will put a dot on the second one. Then the sliding window doesn't just jump to the next two. It actually goes TG, that's the first one. Then it compares GC. If GC matches GC, then you put another dot. Then a CG. So basically it's doing these two against these two. Then slide one over these two, slide one over these two. So then this thing here is a pattern that is inside of here, except that in here there are so many hits that it's harder to see that this is in there, right? So that's why we say, so this is somebody talking and you want to hear them, and this is like a concert in the background, and it's hard for you to extract the message. So now it's like, oh, there's a diagonal, cool, but then there are these parallel lines. Now what we want to see is, what do parallel lines in dot plots mean? So here, I made it in red for you to see. What do you guys see? What do the parallel lines mean in a dot plot? Yeah, good job, Alexis. Yeah, they're duplicates. So in this case, they're almost tandem. There's an extra A in between. But sometimes in DNA, you're going to have a GATT and then no A there and GATT. I just put an A there so that it will be easy for me to find my the duplicate, because otherwise this would all be in red and it wouldn't be as easy for you to find. But yeah, so the parallel lines to your main line, they show the duplications. So now you know, parallel lines show duplications in the sequence. Let's see, a line that this guy is orthogonal, like a 90-degree with the main, that would show you an inversion. And then there is something else for indels, which I think I showed you in the exercise that we're gonna do. So this is a picture of a paper which shows there's like 240 nucleotides aligned against another 240. The numbers don't really have to match. They often don't, but in this exercise they do. So this is just, they're comparing protein, and I know that because this says residue, and residue is another name for amino acid, which is part of protein. So Ken, do you have a question?
So I'm just a little confused. So the diagonal line that's straight down the middle, that's the line that was made when you compared each letter like 1 to 1, but then the other like duplicate lines, that you add those in addition to the main line to show that there was duplicates in the two sequences, correct?
Right. So in this case, the sliding window, in terms of if I compare 1 to 1 or 2 to 2, is not important. I just wanted to tell you that for DNA, we have to use a word size larger because DNA only has 4 variants, and you want to avoid having too many matches that are not meaningful. But the straight line down, and whether or not the slope is like this or if it's up, doesn't really matter. It depends on how the sequence was written. Like maybe it is written this word read from here to here, and this word read from here to here. But if you had written the word down up, this diagonal will be up. So whether it's up or down doesn't matter. What matters is if you see parallel lines to your main diagonal, and if you see an orthogonal line interrupting this diagonal. So these lines here, they were not made because of changing the window size. They appear because there's a duplication in that sequence. Yeah, so did that answer your question? All right, well, thanks. So here, this is, you guys are going to run this, as I was saying, you know, the line, what's the slope depends. In this case, they wrote from left to right and then down up. But here is a gap, which if you are looking at the sequence, this letter matches that, this letter matches that, and then no. There is actually a small gap here, but I'm only pointing to the bigger gap here. So this gap here is sliding up, and this one is sliding to the right. So no matter if it's dislocated up or to the side, that gap here, what's the word that biologists give to gaps? So now a computer scientist looks at this and says, oh, there's a gap there. And you're like, I know what we biologists call a gap. Right, yeah, you could say deletion, you could say insertion, or you could be more complete and say indel. Good job, guys. Yeah, you are learning, I'm so happy. So yeah, there is a gap there. That means there was an indel in this sequence. And here's from a book for like teaching bioinformatics. And this is showing the human genome when there was the draft of the human genome. And see, like there were large inversions, which means they're comparing the near complete with the draft, because we never know, it's not like fully complete ever. I think we're in iteration 8 now. You will learn what that means. But this means that when we initially assembled the genome, we thought the letters had some orientation. But then as we got more sequence, remember how is when you increase the sequencing depth, we call it sequence more chunks. It's analogous to that phrase that we kept picking up pieces of it, the chocolate, dark chocolate, whatever. The more fragments I picked and I read, the more information I gained. So when they sequenced more, they realized that they had assembled something inverted. So these guys are the inversions, and then these are gaps. So there was like a large gap here. And even like in parts like this, they look, straight line. That means we did a good job here. There's no change between the draft and then when we sequenced more. But when you look up close, there's like, all these little pieces that there were inversions. And sometimes inversion doesn't matter. As you will learn, maybe it doesn't impact the phenotype. But if it actually, other inversions actually could be a genetic disease. But in this case, it's not that one's a normal phenotype and the other one's not. This is just, we had some draft and then we sequenced more, and this is us trying to close that gap, but there are a bunch of duplications or inversions. So like little duplications, little inversions. So we're actually going to do BLAST when we do the BLAST exercise, but like for now, what I'd like you to do is grab this paper that you may have printed, and if you didn't, you're going to do that on the screen. So you already have some clue on what you are doing, and it's mostly because I feel like you learn more if you are doing it rather than watch me do it. But I'm actually pleasantly surprised how much you guys learned, and from just me showing you something already ready. Let's see, yeah. So this sequence is something that I came up with. Like there was this read and this read. And I wanted you to assemble them. And my gosh, assembling this would mean that you have to align this to this, and aligning them just by looking is crazy. So what you would do is you're not going to, but if you're a computer, you would put this on one axis, this on the other axis and make a dot plot. So here's the dot plot. I have ABCD, GB, whatever, and then like, oh, look, diagonal line. That means that this part of the sequence is the same as this part of the sequence. The BCD is the same. All right, so here you are going to fill out on a window size of 1 every place where these guys match. So there's a I love Dr.T compared to itself, because I want to show you what pattern emerges when you compare something to itself. So let's see, I'll give you, I don't know, 3 minutes. And if you don't finish, it's okay. It's just kind of fun. It's like the bioinformatics version of Sudoku, but much easier, much more mechanical. And now maybe out of curiosity for you, this is one thing that Word is doing. You know how it highlighted here? It's saying, hey, repeat it. So Word already found my duplication here in my sequence. And when you submit, you know, when you do the safe assign, when you submit some text or whatever on Scholar, it is doing this alignment. It's aligning your work with a huge amount of work that submitted before. And I don't know how it can compare to everything ever, but that's how it finds when you copy and paste something. It's making these dot plots. All right, so let me show you what happens. There you go. You probably could predict that by now. You have your main diagonal, and then you have these parallel lines, which are showing to you that the word love appears twice, right? So you have that duplication. All right, so then there is a love you with love annoying you. And if you were to fill this out, you already could predict, okay, this has a chunk of information, the love annoying you has information that the love you doesn't. So when you fill this out, you'd see a pattern. So you're gonna go, okay, the love and love are the same thing. They'll make a diagonal. The you and you are the same thing. They'll make a diagonal. Might have some spurious matches. If you're more diligent than me, you might find that the letter O repeats quite a bit, and you're going to find spurious matches. But that annoying part is our indel. So then it would look like this. Super clear, right? But so you have a diagonal, another diagonal, and you have this other diagonal that is interrupting that, and it's orthogonal to it. Not quite, right? But it follows a different slope than that. And then there are the spurious matches. All right, I think there is one more, and then I'll give you actually more time for that one. Because this one, you have to fill in, how do you do this alignment? So 10:45, we're going to look at that.